So enzyme promiscuity. This is the new terminology for advanced enzymology, but itself it indicate it is used in many ways. You can say it is the ability of enzyme for multiple functions or multiple activities. It catalyzes multiple reactions, but itself often unrelated chemical reactions. So promiscuous enzyme can accept alternate substrate. They can utilize another ways of substrate or alternate ways of substrate too and perform thereby additional reactions, and that are as per the enzyme name it's not having a primary functions. So name itself, enzyme promiscuity doesn't have a primary functions, but it has a multiple function. It can utilize alternate substrate. This is the characteristic. Now these property can, where it is utilized in human or in evolutionary biology, you can find a certain species having a same chromosome number, but it can survive in the harsh or adverse climatic conditions. For example, rice or wheat, same chromosome number, diploid, hexaploid, or tetraploid. They can grow in the colder environment, hotter environment. The enzyme function is same, even though they are surviving that. So these property can provide evolutionary advantages by allowing the enzyme to acquire new functions without, now remember these blue lines, without the need for de novo gene duplication and evolution. As I said, enzyme promiscuity is thought to be important mechanism for the evolution of new metabolic pathways. It is not possible, but now we can understand that these pathways is improving due to genomics, proteomics, metabolomics, and more number of instruments we came. Therefore, we are now understand what is enzyme promiscuity. It has evolutionary or it can create a new metabolic functions also. And as I said, it can survive in any environment with these properties. So adaptability to the changing environmental condition, it is major functions of enzyme promiscuity. So I will give you an example of enzyme promiscuity. Before that, one of the more terminology, extremoenzymes. Extremo means extreme, name itself extreme condition. Thermo. Okay. Extremozymes are enzymes produced by extremophilic organism that thrives in the extreme environment conditions. These enzymes exhibit remarkable stability and activity under extreme environment conditions. For example, extreme temperature, extreme pH, or you can say extreme salinity or extreme pressure condition or a drought condition. These extreme enzyme has attracted significant attentions or significant importance or researcher. They have a potential application in various ways. You can find in the bioremediations. You can also find in the various process for biofuel productions. And extremoenzyme can also used in the genome engineering and food processing too. So study of extremoenzyme provide a great potential or insight into a molecular adaptations, and that allows enzyme to function extreme conditions. So before these extremoenzymes and their example, we will understand enzyme promiscuity. I have put it that. So we will start with that. Now, mechanism of enzyme promiscuity. To understand this, you require three things, or to various mechanisms of that molecular basis of these enzyme promiscuity includes every enzymes having active sites. So without, as I said, promiscuity means it gives you a multiple functions or multiple reactions. It means there is a flexibility at active sites or other than active site can. So some enzyme have flexible active sites that allow them to accumulate structurally diverse substrates and leading to this enzyme's promiscuity. Then gene duplication and divergence. As I said, it is not giving you a gene duplications. But now I am giving you here one point gene duplication and divergence events such as gene duplication followed by the divergence can lead to a emergence of promiscuous enzyme with altered substrate specificity. With altered altered substrate specificity. Now, as I said, the enzyme promiscuity can change with the extreme environment conditions. Why I have given you extremoenzyme activity or extremo enzymes before enzyme promiscuity. Promiscuity, here you will find a conformational dynamics. These three mechanisms of enzyme promiscuity. This changes conformational changes that enable catalyze secondary reactions and showcasing or promiscuity. Okay, catalytic activities, multiple activities. What is the significance of these enzyme promiscuities? There are four. One is evolutionary biology, protein engineering, drug discovery, and biocatalysis. So you will find understanding this enzyme promiscuity has a several implications or several advantages or several significance that in the pathways of the enzyme and their acquisition of certain new function over the period of times. Now protein engineering, this is another importance of these enzyme promiscuities that can aid or design or engineering or novel functions with altering the enzyme activities. You can utilize these protein engineering, as I said previously, in the industries as well as you can apply in the biological or biotechnological application too. Now, drug discovery. One of the major important significance of drug discovery that can lead to a development of novel therapeutic agents or novel therapeutic drugs or targeted drugs can evaluate with this enzyme promiscuity because of multiple function, because of multiple aids or chemical reactions. Now biocatalysis. No name itself indicate that ability to catalyze the but diverse reaction and expanding the scope of synthetic chemistry. So you can utilize for that also. These these are the example of enzyme promiscuity. Cytochrome P450, aldolase, beta lactamase, and carbonic anhydrase. So these enzyme cytochrome P450 or 450 is known for its substrate promiscuity that allowing metabolites a wide range of compound including drugs and xenobiotics. Then another example of enzyme promiscuity is aldolase that exhibit additional reactions beyond their primary aldol condition condensation activities. Then third example is beta lactamase that demonstrate a hydrolyzing variety of beta lactams that is antibiotics. Okay. Similarly, carbonic anhydrase that is also found in the plant and certain carbonic anhydrase isoform display chemical reactions like enzyme promiscuity by catalyzing not only a CO2 hydration but also it gives ester hydrolysis reactions. So enzyme promiscuity in the plant, you can say that it is the ability of enzyme to catalyze secondary reactions other than the or in addition to primary functions. These phenomena you can observe in multiple metabolic pathways. And these are the examples cytochrome P450s that already been taught to you. Now UDP glucose dehydrogenase. It is also an enzyme promiscuity for in the synthesis of cell wall polysaccharide in the plant. It has been found to exhibit promiscuous activity towards the sugar nucleotides, contributing to the biosynthesis of glycosylated compounds. Another example is isopropyl malate dehydrogenase or dehydratase. Sorry, dehydratase. These dehydratase involved in the biosynthesis of leucine in the plant. Now, what is their other promiscuous activities? That towards the different substrate, it indicate a potential role in diverse metabolic pathways too. So, other than in the biosynthesis of leucine, they are involved in isopropyl malate dehydratase. Then PAL. PAL means phenyl alanine ammonia lyase. It is the key enzyme you will find in the secondary metabolism phenylpropanoate pathways where it catalyzes the deamination of phenylalanine to cinnamic acid. So, in phenolics pathways, however, it has been reported to exhibit promiscuous activity towards the aromatic amino acid, expanding its metabolic functions. Glutathione S transferase. It is also a diverse group of enzymes involved in detoxification process in the plant. Apart from their primary roles in conjugating a glutathione to xenobiotics, some isoform also having some promiscuous activities toward the endogenous substrate. So thereby it contributing glutathione S transferase to redox regulations and signaling pathways. Now, alcohol dehydrogenase, aldolysis, lipoxygenases, these are the example of other than their primary functions. They have revealed the promiscuous activities. So you have to remember these examples. What is enzyme promiscuities? Mechanism of enzyme promiscuities and metabolic versatility and adaptability of plant systems. This is all about the enzyme promiscuity in extreme enzyme. We will complete later on extreme.